So our vision for families SCN2A is a world with effective treatments and cures for all SCN2A patients. Stefan did an amazing job of articulating all the steps that it takes to get from an idea and a concept to an approved drug and therapy. And this is articulated in this figure here of just a lot of steps along the way. And each step are different decision points, ideas, and discoveries that have to be made. And these start with discovery and preclinical, and then clinical trials, and then an approved drug. And each of these can be very challenging and require a lot of minds and thought in order to solve these problems. Oop, I think we're missing a slide. That's okay. So our research roundtable is specifically designed to help solve some of these gaps. So we bring in the best and the brightest minds in the world within the SCN2A space and within the drug development space. And it's a forum for thought, conversation, and networking around these key thoughts and ideas. And we presented on each of these topics on discovery, preclinical, clinical trials, and then approved drugs as well. So in regards to preclinical studies, we had talks on characterizations of learning and memory deficits in a rat model by Dr. Michael Coulter, our last year's Action Potential Award recipient. Stefan mentioned vectors and new different types of gene therapy that are in development. Dr. Yu talked on a new different type of gene therapy that is big enough to encapsulate the large gene that is SCN2A and the work being done there in Dr. Yang's lab. We had Dr. Roy Ben-Shalom talking on really in silico-based computer models of looking at SCN2A, which if validated and proven, could allow us to really look quickly in a high throughput fashion at different variations within SCN2A and allow us to get to answers faster and on bigger network level problems. We had SCN2A splice variants, which is a specific type of variant within SCN2A that typically manifests as a loss of function. And the work being done in Dr. Yang's lab is building out therapeutics specifically around splice site variants. And he's been taking cell lines to animal models and then specifically developing gene therapies for these type of variants. We've also been looked at, so SCN2A, it lives on a lot of different parts of the neuron, and it has to go from where it's produced in the ribosome to those different spots within the neuron, and then it has to stick there. And prior to recent work, it hasn't been known how that's been done. So work done by Dr. Paul Jenkins was talked about how some of that process is being looked at, and then the implications that are downstream of that. And then we had a detailed workshop where all the different stakeholders within our research environment workshopped on additional gaps that exist and ways that we can go faster to get to these treatments and cures for our community. In preparation for clinical trials, we learned a little bit more about how to reverse engineer a drug label. So what is a drug label? It's that little paper form that we get with all of our children when saying which kids and how to treat and how to dissolve in water, all those nuanced steps. But a lot goes into that drug label, and we want to make sure that when drugs are approved, they're approved for all of our children, not just subpopulations. So we want to be thoughtful on how our entire ecosystem is thinking about that. And then we had a conversation about how we should be looking about that and thinking about that with a big panel discussion. We then had Dr. Jennifer Panagonias talk about innovation in SCN2A trial design. Those phase 1, phase 2, and phase 3 trials, there's a lot of elegance and not every trial is the same. And being thoughtful on how you design those clinical trials allows faster time to market and also a broader label when drugs are approved. And they can also increase the probability of success of that trial, and then also increase the probability of success of us getting new treatments for our children. And then we had a panel discussion around barriers that exist in drug development from all of our industry partners that are actively working on developing these drugs. So they gave their perspectives of their wish list of ideas and concepts and models that they wish they had in order to move faster within the context of SCN2A. And then we talked about clinical needs. So our children, and there's still big gaps in the clinical care for our children. And you're going to hear a number of talks from different caregivers and their shared experiences. And I think every one of the caregivers in this audience, and I know I do with my daughter Harper, that there are barriers that exist in optimizing clinical care for our children because they're complex. So we talked about a panel discussion with some of the best clinicians in the field of SCN2A, talking about how do we take the ideas that are in all of their heads, which are kind of the best and the brightest, and disseminate it to all of our clinicians in order to raise that level of care, and how we can do that in a short amount of time. And then we had a provocative study by Dr. Louise Taranoff and their project on videography. And it's an interesting project that is taking video from all of our children in kind of a natural history study, and allows you to kind of characterize your child as they live in different parts of their life. And why that's important is it can aid in drug development, and those videos can then help new families that are diagnosed with SCN2A recognize symptoms that maybe they're seeing your child move a little bit different, that looks a little strange. It's hard to communicate that to your clinician, and then it takes a very long time between that incident that's maybe happening randomly and then getting the diagnosis with your child. Capturing these on video could be a way for us to streamline and get diagnosis quicker and have a more proactive approach to SCN2A management. And we had a lot of fun. You can see Dr. Kevin Bender, who's been source of a lot of pictures on these slides so far, giving thumbs up and also doing a sprint up to the stage. So we hope that we'll have a lot more fun during this conference. But we kicked it off well with a research roundtable yesterday.